yeah, we'll get going from here. So welcome everyone out there, those with videos on and those without videos on. But welcome to this webinar about probiotics. And today is the first time that we're doing a little bit more like product commercial webinar. But I got quite excited about this webinar because there's actually some publications that you can find in PubMed on some of the products here and some of the probiotic strains that Dr. Bayne is going to present you to. So I have asked for Dr. Bayne to make a more educational webinar, and I have already sneak peeked on some of the slides, and it looks as if you are in for a treat in regarding to probiotic knowledge. So I'm Anne-Catherine, as you all know. I'm the co-founder of Nordic Laboratories and DNA Life, a group of companies dedicated to change healthcare through functional medicine and personalized medicine. Our next webinar is the Nordic Clinic series, and it's on thyroid testing with Graham Jones. And remember to sign up for the invites that's coming out. It's on Thursday, the 17th of June at 4 o'clock Copenhagen time. So 3 o'clock England time, and yeah, wherever you are in the world. Remember that lab tests and also supplements you can purchase in your practitioner profile in VMS. You can order for both yourself and your patients. And as always, we will mail out the slides to you and the recording. And I will add a few links to the PubMed publications and what else I can find out could be fun to send out to you. So before I just shortly introduce Dr. Bayne, then I just want to say that we are still working on the new VMS platform. It's very exciting, but it's really beginning to look good. We keep testing and we keep getting new ideas, but soon, all good things for those who wait. Anyway, the star of the show today, Dr. Bayne, who is the president of Microbiome Labs. He's got the logo behind him. It looks very nice and colorful. And his background is in, he's a chiropractor, but he also holds a degree in human biology. And he still sees patients, and I believe he's done it for more than 25 years. Is that correct?
Correct, yes.
Yeah. So, and then he keeps on educating, and he's a really good educator. So he keeps educating practitioners in new products, and of course, finding out what new products there is to work with and developing those and well. So he's got a really interesting background, and I'm so much looking forward to sharing him with you today. So remember to put your questions in the Q&A. Because I will keep them, and then at the end of the talk I will put up some of the questions, and we can have some discussion or whatever happens, you and I, but write them and write to each other and answer each other, and what have you. So on to you, Dr. Bayne.
Thank you, Anne-Catherine. Appreciate it. It's great to be here with all of you today. I'm very excited to share this information with you. Before I get started, I'd like to just say one thing. I'd like to say that the intention of this talk is to introduce you to new science, new information on the human microbiome and probiotics. The talk is not intended to make you feel like you've been doing things wrong up to this point, or that the other products that are in the marketplace are not good or anything like that. That's not the intention. But when you talk about something, you have to compare it to what's already in the marketplace. And so oftentimes I'm told, don't say bad things about other probiotics, and that's not my intention. What I'm going to compare what the science of probiotics is all about, and present that to you. But I'm here more to add to your toolbox, not subtract. So the intention here is to introduce a new concept that you can add to what you already do. So I just want to say that up front so that we're clear, and I'm excited to present this information to you. So let's get started. So the basis of the information that we're going to talk about today comes from the Human Microbiome Project. And so the Human Microbiome Project was a study that was commissioned by the National Institute of Health here in the US. But it was commissioned in the US, but however, it was a worldwide consortium of researchers and teaching institutions. So there were over 200 researchers that were part of the Human Microbiome Project from 80 different institutions worldwide. And the reason they did it was because the human flora had never been completely characterized. To actually take a human being and look at all of the bacteria that live in and on their body and to understand that. And that had never been done. And the reason it had never been done is because a vast amount of the bacteria that live in our bodies are obligate anaerobes or strict anaerobes. So it's very hard to use typical microbiology plating methods and different things like that to see what kind of bacteria live in us. So it wasn't until we got the metagenomic technology in place. And so all the people that did the Human Genome Project, when that was done, they all shifted over and started working on the Human Microbiome Project. And so let's look at some of the facts from the Human Microbiome Project. So we are more bacteria than we are human. So we have about 10 trillion human cells, but we have over 100 trillion bacteria that live in and on our body. There are over 1,000 different beneficial or commensal species in the gastrointestinal tract out of 35,000 possible. That's a much bigger number than we thought. And so to think that there are 1,000 beneficial bacteria in your digestive tract, yet you're supplementing the same 5 to 10 bacteria, we'll get into that, the science of that and the understanding of that, and does that really make sense? So the other thing is that the bacterial genes outnumber human genes 150 to 1. So humans have about 25,000 genes, yet there are 3.3 million microbial genes, and the crosstalk between those genes and our genes and the interactions that they have and functions they have within our body are very unique and very interesting, and we're still discovering a lot of that information. No two individuals have the exact same composition. So they've looked at twins. They look at twins who live together and eat the same foods, and still you can tell them apart by their microbiome signature. So the phylum levels are very similar, but when we break it down to the species level, it's completely different and very much like a fingerprint to that individual. So it's often been taught in schools and in conversations when you're talking to microbiologists that the intestinal microbiota is just this giant cauldron, like a giant vat that ferments everything. And that's not true at all. What we're seeing is that it's very intelligent, very specific. We see different kinds of bacteria in different areas of the microbiome. We see different bacteria even in the different, as we go from the epithelial layers into the lumen of the intestines, we see different bacteria. And so this is an interesting slide, and this is just kind of an overview of some of the different things that we're dependent on our microbiome for. Some of the things that we understand, obviously, the digestion and absorption of nutrients, okay, that's obvious, but anxiety and stress behavior comes from the microbiome, and that's due to the production of serotonin, dopamine, and norepinephrine, and the repression of corticosterone and adrenocorticosterone. So anxiety and stress, a lot of that lives or comes from the microbiome. Our metabolism and appetite. So the changes in ghrelin and leptin are associated with the microbiome. And so alterations in insulin levels, that all comes from the microbiome also. So the immune system, the microbiome is hugely important for our immune system. And it's also important for the production of pheromones and mating and species recognition behavior. So, and even we can even go as far as saying that alterations in androgens and estrogens and prolactins lead to gender changes and different things like that. So what the gist of this slide is meant to show you is that there's a lot more going on in your microbiome than just the digestion of food and the absorption of the nutrition from that food. So where does our microbiome come from, right? So what we have is we have this seeding that happens while the baby is in utero. So we have data that shows that macrophages from mom go into her microbiome. They grab on certain bacteria in her microbiome, and then they bring it into the amniotic fluid, and then that bacteria circulates as baby drinks the amniotic fluid. As the amniotic fluid goes into the baby's digestive tract, then the bacteria start to initially seed the baby's microbiome in utero. We then have the birth canal. As a baby travels through the vaginal tract, there's a huge inoculum of bacteria that enters the baby at that point. And then in the first year, 2 years of life, there's breastfeeding. There's over 600 different bacteria in breast milk. And then there's the cuddling and interaction with mom and dad. And at 2.5 years of age, 838 days roughly, baby has their adult microbiota. And so this is fascinating information. And if you look at the slide here, what you see is that initially babies, the bacteria in the baby's, in and on the baby's body kind of represents everything that is coming from mom's vaginal tract. But then over time, the skin, the mouth, the gut all start to take hold and to look more like an adult microbiome and to be the home of the bacteria that are healthy in the mouth, on the skin, or in the gut. And so this is how we get our microbiome. After 838 days of life, roughly 2.5 years, the only thing that happens after that is that the environment influences our microbiome. And so this is more of a conditioning of our microbiome, not necessarily a seeding. And but we'll go through this in more detail as we go forward here. So any diversion from this natural process leads to disease. And that includes C-section, right? So what we're seeing in a C-section baby is that baby's microbiome looks more like the doctor and nurse's hand bacteria, the skin bacteria, than it does mom's vaginal bacteria or mom's gut bacteria. So that early diversion from this natural process, a C-section, leads to dis-ease and potentially disease down the road, but it leads to dysfunction and potentially disease down the road. So our goal is to create a diverse microbiome. We know that. We know that a diverse microbiome represents a healthy microbiome. So that's our goal, right? We know that, and we know the seeding part, right? But currently, the probiotic market is attempting to reseed. Everything that we do in the probiotic industry up to this point represents an attempt to reproduce what's happening between mom and baby. It's a reseeding of the microbiome with good bacteria. And so what we're going to do is we're going to look at the science of that, and we're going to look at some potential other options. So when we talk about reseeding of the microbiome, it's great when we're talking about a patch of grass that's missing in your front yard, but we're going to talk about some issues here. So one would be survival. Do these bacteria actually survive, get into your digestive tract, and colonize your microbiome? We'll talk about the science of that. Look at fitness. Once they get there, can they do anything? Are they functioning? Are they doing probiotic duties in the microbiome? We'll look at the science of that. Is it natural? Caveman didn't have a refrigerated section in his health food store. So where did he get 50 billion CFUs of lactobacilli and bifidobacteria on a daily basis? So we'll look at this natural, is it a natural strategy here? And then we'll look at the quality of some of the products that are in the marketplace and whether or not that holds up to what we want to be doing with our patients. So this was a study that was done here in Chicago. And what we did is we looked at 4 different products. We were looking at survivability through a gastric model. And so we used a USP gastric survivability model. And so what that means is that we used a gastric solution that was at a pH of 1.3, and then we allowed these different products to sit in that pH solution for 2 hours. And then afterwards we added bile salts, and I'll explain in a little bit why we did that. But that is a simulation of your digestive tract. And the products that we used were Ultimate Flora, which is the best-selling probiotic in the retail markets here in the US. We used Chobani Greek yogurt, and we used Dannon regular yogurt along with the Megaspore Biotic. We did this test. And so what we saw was that on the initial testing, we had 100% of the probiotics were identified. So the probiotic yogurt, believe it or not, had 5 billion strains in it, which was interesting. We later found out afterwards that Dannon adds strains back after pasteurization and homogenization. So that's why there were 5 billion strains in there. So we were very excited to see live bacteria in the Dannon yogurt. The Chobani yogurt had live bacteria. It was only about 400 million. It was much less than the yogurt did, and much less than we're typically used to seeing when we're supplementing lactobacilli. But nonetheless, there was some bacteria in the Greek yogurt. In the Ultimate Flora, you see on the label, it said 50 billion. So when we analyzed the Ultimate Flora, there were actually 230 billion CFUs per capsule. And so you might ask yourself, why would a company put 230 billion CFUs, but only put 50 billion on the caps, on the label? And the answer to that's quite simple. It's because the people who own the company know that the product is, there's attrition in the capsule on the shelf. They know that the product is dying on the shelf. And they know that they have to have 50 billion by the time of expiration. So in the US, there's a lot of suing. If you say something on a label and then that proves not to be true, then you can find a greasy lawyer in California that'll sue that company and give you money. So in order to prevent that, they put overage in their probiotics. Now, in a lot of parts of the world, you're not allowed to put overage. And so as a result, you're not allowed to, you don't see those products very much in the marketplace. So Scandinavia is one of those areas of the world where you have an under and an over. You can't go over. If you say 50 billion, you can't go over 70 billion. And so these products are not available in Scandinavia for that very reason. And so what we saw was, on initial testing, everything was fine. We dropped these products into the bile salts, into the pH solution, the gastric solution, for 2 hours, and then we added bile salts. The reason we added bile salts is because some bacteria love acid. Lactobacillus acidophilus, it loves acid. So it's fine in a pH solution for 2 hours, but the second it hits bile salts, it's dead. And so a real gastric simulation study, when you see other companies that do these types of studies, frequently they'll say that the pH was at 3.5, and that there was 100% survivability at 3.5. Well, if your stomach pH is 3.5, you're sick. You're not well. And lots of bacteria are probably sneaking past your stomach acid. So this is a legit test. And when we did it, what we saw was complete destruction of the bacteria in the 3 products that we tested. The 230 billion CFUs of Ultimate Flora were completely decimated. There was no bacteria shown afterwards. Same thing with the Chobani, same thing with the Dannon yogurt. The Megaspore had 100% survivability. The reason is because it's a spore, and we figured out how to keep it 100% spore form. So it's able to survive in a harsh environment like the stomach acid. And so this was an early study that we did. So let's look at fitness, right? So all right, let's forget the fact that it looks like a lot of these bacteria don't survive. Let's forget that for a second. And let's pretend, okay, they get there. Once they get there, well, can they function? And I would question that because one, you're looking at an aerobic bacteria that's trying to live in an anaerobic environment. And so when we grow these bacteria, we're growing them in fermentation vats in aerobic conditions. We got them in a capsule and they're alive. We open the capsule, we can see them under a microscope. They're moving around. If they're moving around in an environment where there's oxygen, they're not anaerobic, right? Yet the vast majority of the bacteria that live in our microbiome are anaerobic. And so we would question the fitness. We would question whether they can actually perform a bacterial function when they're in the microbiome. And so, and the other thing too is, do they have binding sites? I can't look at you, I can't muscle test you, I can't, take your blood or take a stool sample and see exactly what bacteria you need. Like I said, we're looking at 1,000 different bacteria. And so how can I figure out exactly what bacteria you need? I don't have a method for doing that. And so there's not an acceptable method for me to be able to say, oh, you need Lactobacillus rhamnosus. I don't know that. I can't evaluate that with you. So it's very difficult to know whether you have the binding sites that would be necessary for you to take on that bacteria. And then, is it really natural? Well, where did caveman get his bacteria? Did he get his bacteria by eating the digestive tract of dead animals or animals that were freshly killed? We see that in some more like the African tribes. When the guy kills the animal, the first thing he gets to do is he gets to eat the intestines. Sounds pretty gross to those of us, but that's one way that the humans were getting that inoculum, but not a very common way that we would be getting bacteria in our digestive tracts. So the study on the fitness is an interesting study. There's a comparative study that looked at bacteria's ability to bind, right? Okay, we can get the bacteria in the digestive tract, but can we get it to bind to receptor sites? And so this study showed that the best adhesion in a Petri dish was 38% by Lactobacillus reuteri. Second best was 24%, and the rest was down there. If we can't get it to bind in a Petri dish, how do we get it to bind in a digestive tract where we've got, trillion of 100 trillion other bacteria competing for space and competing for food and things like that. So it's kind of similar to, where does, we can seed the microbiome in a similar way that we can grow fish in our garden, right? It doesn't work. And from the natural standpoint, when we're looking at, okay, I've got this Lactobacillus bifidobacteria product. It's got 10 strains in it, and it's got 100 billion CFUs, and I'm taking it every day, and I'm taking it every day. Well, if it actually did what they say it does, you would be creating redundancy in the microbiome. You would be crowding out those thousands of species and really only making 10. And so thank goodness that's not the science of Lactobacilli and bifidobacteria. We're going to get into that soon. I'm not saying those products aren't good products. That's not what I'm saying. What I'm saying is that the reasoning and the bridge that we've built is dysfunctional. The story that we tell ourselves that these bacteria go in and they live happily ever after in our digestive tract, that's not proven by science, right? That's not supported by science. So that doesn't mean they don't have benefits. So we're happy that the idea in our mind of these bacteria going and colonizing in the microbiome, that's not supported by science. Because if it was, we'd be hurting people. We'd be creating redundancy in their microbiome. We'd be reducing their immune function and their ability to absorb nutrition. So there've been a lot of research around the quality of probiotics also. So several documented cases, especially in the EU, of products that were incorrectly labeled with the wrong strains, products that contain markedly reduced numbers that were claimed. We see that across the board. And then there were also the presence of other bacteria that were not on the label. So In some cases, these bacteria were pathogenic bacteria. So, when I'm speaking in lectures where I'm talking to other people in the industry, I make a big focus on this because this is where we're going to run into trouble. This is where problems are going to arise. And so this was a study that was done at the end of 2015 at the University of California, Davis. And what they did is they took 16 different probiotics from different shelves, stores throughout California, and some that were online. And they specifically took bifidobacteria products. And the reason they did that is because these are the probiotics that we give our children, specifically infants and babies. And what they found is of the 16 products, 15 of them didn't meet label claim. And only one met label claim. And that one that met label claim was one strain in a very special capsule and all that other stuff. So what they were able to show in this study is that the probiotics that we give children don't meet label claims. And that's when the authorities are going to get involved, and then things are going to get ugly. So like I said, when I'm speaking to people in the industry, my drum that I'm beating is you've got to work on meeting label claims, and you shouldn't be selling products if you know you're not meeting label claims. And if you're selling a product with 230 billion in it, but you're only advertising 50 billion, then you know you don't meet label claim. And I'm hoping that this doesn't hurt the entire industry because of a couple of aggressive companies that are trying to make a buck. So clearly I've been able to show that with lactobacilli and bifidobacteria-based products, there's some issues. There's questionable whether they survive, questionable whether once they get there, they can do anything. It's definitely not natural. And there's clearly quality issues. Then why do they work, right? We all know they work. We all know, we've all had experiences with lactobacillus and bifidobacteria-based products that work, and we've seen benefits. We've seen them change people's lives. How can that be, right? So I'm not here to dispute any of the research on lactobacillus and bifidobacteria. I'm actually here to support it. What I'm here I want to have a conversation about is the story, the marketing story that the companies tell us, right? So an example, Lactobacillus rhamnosus GG, fantastic information about that product in the treatment of chronic urinary tract infections in women. I do not dispute that research in any way, right? That's great research. But then when you ask so I had a salesman come to my office with that study. Look at this. I was like, well, I've got some patients with chronic urinary tract infections. I'll use that. By the way, I'm like, what's the mechanism of action? Well, you swallow the Lactobacillus rhamnosus GG, it goes into the digestive tract, and then it makes its way to the vagina. What? How did it do that? Right? There's no tract from mouth to vagina, right? So the bridge of how the product works is not supported by science. It's marketing information, right? That's the point I'm making. There's clearly benefits to these lactobacillus and bifidobacteria products, with certain caveats, right? So let's look at some of the research. So here's some studies that showed that the dead bacteria, the dead lactobacillus and bifidobacteria products functioned as well, or in some cases even better than the live bacteria. Okay, well, that's interesting. So when we look at the World Health Organization's definition of a probiotic, what we have on that definition is that a probiotic is a live microorganism which, when administered in adequate amounts, confers a health benefit to the host. Okay, so therefore, by definition, viability of the bacteria is paramount to the definition of being a probiotic, right? So as a result, the vast majority of probiotics that in our minds that we've used for decades with our patients, they don't really fit the definition of what a probiotic is. What they do fit the definition of is a biological response modifier, or I prefer a metabolic response modifier. A metabolic response modifier is something that goes into your microbiome and it changes a symptom, right? That's what a metabolic response modifier does. And some of them do it very well. And we'll look on this, well, I have a slide coming up where we'll look at that in more detail, but our current strategy of trying to reseed the microbiome has its shortcomings. I would say we should really be looking at the environment. We shouldn't be trying to recreate what nature has perfected. We should be trying to look at what nature still has to offer us, right? We can't repeat what happens between mom and baby, but there is an environment around us, and there are organisms in that environment that can improve the health of our microbiomes, and that's what we're going to focus on. So what are the aspects? What do we need to see from a healthy microbiome? So these are some studies that we're going to show that people that live off the land, people that live a rural kind of lifestyle, they have much healthier microbiomes than people who live in established or Western civilization. So this study showed that the distal gut of Bangladeshi children had significantly greater bacterial diversity than that of US children. Okay, so, you know, US children don't run the risk of getting eaten by Bengal tigers, right? So it's not easy being a Bangladeshi child, I understand that, but they do have much healthier microbiomes than a kid in New York City or than a kid in Chicago, right? They have a much more diverse and therefore much more healthier microbiome. And a Bangladeshi child is living off the dirt. He's eating dirt every day. He's getting in his sinuses. It's everywhere, right? And so as a result, they have a much more diverse microbiome. So this is a study that was done. We have studies that were done in Japan between industrial areas and rural areas, and we have studies that were done in Russia, again in between industrial areas and rural areas. And what we were, again, in those studies, each were able to show that there were much more diverse, much healthier microbiomes in the people that were living a rural, off the land kind of a lifestyle. So what are the requirements of a healthy or natural probiotic? So you have to be able to show that they can survive the harsh gastric environment. We have to be able to show that they can live in the acidic environment of the stomach. The stomach is a barrier to entry. If the stomach allowed any bacteria to just cross over, then we'd all die in the first 10 years of life of some type of gastric infection. The stomach is an incredible barrier to entry. It doesn't just let any bacteria slip by. So a true probiotic has to be able to show that it can survive. It has to be a strain that's found in the microbiota. And not knowing which of those 1,000 strains that people have, it's very difficult for you to be able to say whether the strain exists across the board in everybody. It must have evolutionary significance. If the product needs to be refrigerated in order to be supplemented, then caveman didn't have it, so it has no evolutionary significance. It's probably not part of what man evolved with. It must be a facultative anaerobe. It's got to function in an oxygen depleted environment because that's what our gut is. So if it can't function there, it's useless. It must have a biphasic lifestyle. If we're able to supplement it, then it has to be able to function in our environment. And if it's going to be a probiotic, then it has to have probiotic benefits in our digestive tract. So it has to have a biphasic lifestyle. And obviously, it must show clinical demonstration of safety and efficacy. You'd be surprised how many bacteria on the store shelves don't have safety and efficacy data. So what I'd like to introduce you to are bacterial spores, and specifically Bacillus spores. So Bacillus spores are the most widely studied and most widely used probiotics outside of the supplement market. They were the first commercial probiotics, and they were the first prescription probiotics. So in 1958, Sanofi Aventis created Enterogermina, and Aventis created Bactisubtil, And so these were prescription probiotics in the late 50s, early 60s that fell off the radar because of the advent of antibiotics. We see spores being used extensively in agriculture and aquaculture. Here's a few of the brands that are out there. So in agriculture and aquaculture, it's very easy. The farmer, he's able to say, I have this much input, I get this much output. And so he tries a probiotic. If it doesn't increase his output, he doesn't use it because he can't justify increasing his input when he's not getting more output. So the only probiotics that are used in agriculture and aquaculture are spore-based bacteria, because when you put them in, you get more out. So the most widely used and well-studied strains in humans are Bacillus subtilis, Bacillus licheniformis, Bacillus coagulans, Bacillus clausii, and Bacillus indicus. So key features of a spore. So when the bacteria is in an environment that's not conducive to it, whether there's no food, the temperature's really hot, there's a low pH, there's antibiotics in the area, what the bacteria is able to do, it's able to dehydrate itself and form a very strong outer protein coat. That's the spore. And it's basically in a state of suspended animation, or it's inert. It's waiting to get into an environment where it can thrive. And so if we drop a spore into your stomach acid, it's not coming out because it knows I can't thrive here. There's no food for me. But once it gets into the upper part of the small intestine, it feels that pH change. It feels the temperature change. It comes out of its spore and it goes into its vegetative state and starts to perform its probiotic functions. So we find spores all over our environment, in the soil, vegetation, dust, rocks. We find it in water. We find it in the digestive tracts of everything we've looked at: insects, animals, whales, lizards, humans, everything we've looked at, we find Bacillus spores in the digestive tract, and we find receptors for the Bacillus spores in all the digestive tracts that we've looked at, right? So that's unique. Spores can remain dormant for 250 million years. So the oldest thing that humans have ever found was a spore in a cave in Brazil. So what they did is it's a little Jurassic Park-ish, but it's a fun story to talk about. So they found a fossilized amber, and in the amber, there was a honeybee, an extinct honeybee, a very large honeybee. And so they went in and they opened it up, and inside its stomach, there was some brine. And they looked through the brine and they found Bacillus subtilis in the stomach in spore form. And so they took it out, they plated it, they created an environment that was conducive for the spore. And guess what? It came alive. It came out and into its vegetative state and started saying, hey, there's food here, I'm here. It was 250 million years old. So that spore had sat in a dormant state for 250 million years, and then under the right conditions, it came out. Pretty cool story. Oldest living thing that as humans, we've been able to identify. So there's these ice core studies that they do all around the world where they take these coring devices and they go down a mile and a half into the ice, and they pull it out. And that's meant to show us what the world looked like 10,000 years ago, 20,000 years ago, you know? And so as deep as they go, as far back as they go, every time they pull up these ice cores, they find Bacillus bacteria. So these bacteria have been with us from the beginning of time, since the beginning of man, we can say that. And so we've co-evolved with these bacteria in our environment. They colonize very effectively in the human GI tract, and they've become found to also colonize very effectively in the gastrointestinal tracts of animals that have been tested. They're part of our normal human commensal flora. So they're not a soil organism. They are human commensal flora. That's what they are. Because Bacteria are labeled based on where they function, where they perform their functions. So a soil organism is something that functions in the soil. A human commensal organism is a bacteria that functions in the human digestive tract, right? So spores are commensal flora. We can't say they're human commensal flora only because they function in animals also. So they're commensal flora, but they do function in humans. So they are human commensal flora also. And so they have a long history of use in industries where efficacy is very closely measured. That's pharma and agriculture. They're extremely safe. They're supported as, there's an evolutionary component to this. We can see that caveman had spores in his cave. So those are the basics of the spores. And so what we're going to look at now is how they function. So there's numerous studies and a long history of use confirming the survivability of spores throughout the gastrointestinal tract. Let's take a little journey, okay? You swallow the spores. They're in spore form, 100% spore form. They go down into the stomach acid. They survive through the stomach acid. They get into the small intestine. They come out of spore form and they start to perform functions in the upper part of the small intestine. In the lower part of the small intestine, they resporulate. We don't know why that is, but we do know that that process of resporulation triggers the galt, the Peyer's patches, the immune system of our gut. It stimulates those tissues. So that stimulation of that gut-derived immune tissue creates a modulating effect on our overall immune system. So even though we don't know why they resporulate in the lower part of the small intestine, that's an important component to get that overall immune modulating benefit that the spores give, okay? So then once they get to the upper part of the large intestine, they go back into their vegetative state. They're out of spore form and they're functioning as a bacteria. And so there are studies that show that the spores are well adapted to germinate in the small intestine, proliferate, and then they resporulate in the lower part of the large intestine in anticipation of leaving. So the spores spend 21 to 28 days working their way through your digestive tract. So they are transient in nature, but not transient like Saccharomyces boulardii, right? Saccharomyces boulardii spends 48 hours in your digestive tract. It just basically passes through. Spores spend 21 to 28 days. They're performing functions while they're in there, but they have no intention of staying there. They want to work their way through, perform some functions, and then move on. So Bacillus spores are found in the GI tract of insects, animals, and humans. We've said that already. Studies indicate that the environment is a vector that spores use to move from host to host. So as it resporulates in the lower part of the GI tract, goes into the environment, and then it waits to get into the GI tract of another animal, insect, human, whatever it might be. So spores have been shown to shift the microbiome. Okay, this is an important factor. What we've talked about with the Lactobacillus and bifidobacteria, they're hugely important for very specific things, very specific changes to symptoms, but they do not change the microbiome. And the minute you stop using your Lactobacillus and bifidobacteria-based products, the benefits tend to dissipate and go away. And that's because you're not really making a change. You're treating a symptom, you're not making a change. And so spores actually shift the microbiota. They actually change the microbiota, improve the functional microbiota. So spores have the ability to shift the microbiota in a number of ways. First of all, they can produce antibiotics. So there's a number of very specific types of antibiotics that they can make. So what the spores will do is they use quorum sensing to read the microbial environment. They know who's supposed to be there and who's not. And so when they see a bacteria that's not supposed to be there, they go in and they first try to competitively exclude it. Try to just fight with it for space or food. But if that doesn't work, they'll actually produce antibiotics to kill off the bacteria. And so they also are able to increase They themselves produce short chain fatty acids. Whoa. Yikes. My screen left. Sorry, guys. I don't know what just happened. Shoot.
Don't worry, no stress.
What happened to my slide? Oh, there we go. Phew. All right. So spores actually have the ability to produce short chain fatty acids. And short chain fatty acids are responsible for creating that acidic environment in the large intestine that crowds out yeast and crowds out pathogenic bacteria. So the spores are working from two angles. They're fighting the actual bacteria and yeast that's not supposed to be there. And then they're producing waste products that are actually changing the environment or improving the environment of the intestines, which will then increase the growth of the commensal bacteria that we got from our mom. And so we're working on improving and shifting the microbiome in two different ways when we use spores. So we're not aware of any other probiotic that's demonstrated the ability to fix dysbiosis. So we have studies that show that we can shift the microbiome in 6 weeks by 30%. We can improve the growth of keystone strains that are responsible for producing short chain fatty acids in our microbiome. We could show a 30% increase in those bacteria in just 6 weeks through the use of supplementation of spores. So that's science showing that we can change the microbiome. It's science showing that we're not treating symptoms, we're actually changing the microbiome. Immune modulations. So there's actually research that shows early exposure to Bacillus subtilis by the infant microbiome, early exposure in utero and early exposure after birth in the early stages of life helps develop the human immune system. So without spores, the human immune system has a challenging time developing and getting to the place where it needs to be in order to protect the individual. So spores increase circulating T and B lymphocytes. That's part of what they do when they're stimulating the Peyer's patches in the small intestine. Spores are able to shift the body from a T2 inflammatory to a Th1 adaptive state. This is due to the stimulation of toll-like receptors, interferon gamma and interferon and alpha stimulation. So we're able to shift the body from the body from an inflammatory state to an adaptive state. That's what we want. That's what we're looking for when we're treating patients. Spores have been able to improve pattern recognition and curb autoimmune and allergic immune responses. This is a huge aspect for people with allergies and autoimmune disease. Bacillus spores shift the innate immune response to an adaptive immune response. So this is via dendritic activation. So spores are hugely important for the health of for the development and the health of our immune system. Spores also on their own are like tiny little nutrient factories. This is the part where, like, if at this point you're not already like, wow, this is the coolest thing I've ever heard, this is where will really blow your mind. So the spores, we talked already about their ability to produce short chain fatty acids on their own. So what we've been able to show is that we can increase short chain fatty acids by 40% in 6 weeks. Now that's a combination of the spores themselves producing short chain fatty acids, and as the spores change their environment and improve the health of their environment, the good bacteria that produce short chain fatty acids increase in their numbers. So it's not just that the spores produce 40% more short chain fatty acids. It's the combination of the spores and the return of the healthy bacteria in the microbiome that produce short chain fatty acids. So that's huge because short chain fatty acids stimulate the colonic blood flow. They help with electrolyte uptake. It helps to heal the colon. The colonocytes require those short chain fatty acids to function. But those short chain fatty acids are also important for our metabolism. We've got high concentrations of short chain fatty acids in our microbiome. It increases our body's ability to use fat for energy. That's hugely important. The growth, the increase in short chain fatty acids supports the growth of lactobacilli and bifidobacteria, our own lactobacilli and bifidobacteria, the anaerobic type that we got from our mom. It's also been shown to lower cholesterol and triglycerides, reduce gut and systemic inflammation, and decrease intestinal permeability, which that's our leaky gut, right? So they have the ability to produce all of the B vitamins in their methylated forms. They produce vitamin K2. They produce, and then HU36 produces carotenoid antioxidants. So this bacteria survives through the stomach acid, gets over into your small intestine, and it starts producing RDA levels of alpha carotene, beta carotene, zeaxanthin, astaxanthin, lycopene, and lutein. It's producing these carotenoid antioxidants right at the site of absorption. And so this is a huge game changer in bacteria. This bacteria wasn't discovered until 2010. And it actually is the best way to improve your carotenoid status in your body, because carotenoids themselves are very sensitive to the stomach acid, and they don't survive the stomach acid, similar to the way bacteria don't survive the stomach acid. So when you look at the RDA of beta carotene, 800 milligrams a day of the RDA of beta carotene, that's with the hope that 8 milligrams are going to survive the stomach acid, right? So we're on the other side of the stomach acid, producing 800 milligrams of beta carotene a day. So what we've got is a very unique product from both a microbiome shifting perspective and nutritional perspective. So we've done some research around leaky gut. We'll talk about that, the specific research in a second, but there's plenty of research that shows that toxins that leak from the gastrointestinal tract into the bloodstream create an immune dysregulation and inflammation that's at the root of inflammatory bowel disease, Crohn's disease, ulcerative colitis, every autoimmune disease that we've studied to this point. It's the driver of the inflammation in cardiovascular disease. It's part of the inflammation of cancer, especially when we're talking about the spreading of cancer. And it's part of the dysfunction that takes someone from HIV positive to AIDS and death. All of these conditions that I just mentioned have mounds of research that show that toxins that spill from the gut into the bloodstream and the associated inflammation that causes, that's at the root, either the cause of or part of the pathophysiology of all the conditions that I just mentioned. So if we could treat leaky gut, if we could show that we could reduce the amount of toxins spilling into the gut and the amount of inflammation associated with that, then we basically have a probiotic that should be the cornerstone treatment for all these conditions. So let's look at leaky gut. So on the left, we have a healthy gut. On the right, we have a leaky gut. Let's talk about the difference. Let's focus on the left. Let's look at what pretty looks like, right? So on the top, that's the microbiome, right? The microbiome is sitting on top of that blue area. That blue is the mucin 2 layer. It's the mucin layer. It's the mucus layer that sits on top of the epithelial layer. And the epithelial layer is that, I like to call it that caterpillar looking like thing, that one cell that is separating our environment from our basolateral circulation, from our bloodstream, right? So it's very important that the mucus layer buffers the microbiome away from the epithelial layer. That keeps everything moving through the lumen of the intestinal tract and leaving in the stool, okay? What happens as the microbiome becomes dysregulated, as the microbiome becomes, as we start to see some dysbiosis, some of those bacteria start to erode the mucin layer. They start to eat away at that mucin layer. And eventually they eat away at that mucin layer so much that now the microbiome is laying on the epithelial layer. The epithelial layer doesn't have the ability to say, oh, that's a beneficial bacteria and that's a pathogenic bacteria. It just sees bacteria. And when it sees bacteria, it responds with an inflammatory reaction. And over time, you start to see spacing between the different aspects of the epithelial layer. And so when we look at the leaky gut schematic to the right, we see that the mucin layer is eroded, that the microbiome is touching the epithelial surface. The epithelial surface has become inflamed, and now they're spilling into the circulation. So for a second, let's go back to the left. At the top, you see LPS. It's at the top, lipopolysaccharides. LPS is a normal component of gram-negative bacteria. When gram-negative bacteria die, they lyse, and the LPS from their membrane spills into the area. It spills into the lumen of the intestines. If that LPS leaves in your stool, there's no problem. But if that LPS spills into your bloodstream, there's a big problem. So now come back to the right, and we see that the bacteria is eroded. We see the inflammation, and we see LPS and bacterial byproducts spilling into the bloodstream. We then see a macrophage grab onto that LPS, grabs it at the toll-like receptor. It looks at it, examines it, and then it says, oh my God, we've got septicemia here. And it goes on to produce interleukin 6, tumor necrosis factor alpha, interleukin 1 beta. It starts to create an inflammatory reaction because it thinks you're dying of septicemia, right? This is a low-grade inflammation that happens in some patients 3 times a day because that's how many times they eat. And this reaction, this inflammatory reaction, it's at the root of all those conditions that we just talked about on the previous slide. Okay, so we've talked about it, and now I've given you kind of a visual to see it. So let's talk about our study. So this is the pilot study first, because we didn't know what we had. We met a guy in Texas, and he said, I've got this model where I can study leaky gut. We were like, really? What's your model? Well, what he does is he feeds students. First, what he does, he measures students' blood coming in off a fast. And he looks to see if there's any LPS in their bloodstream. Most people have no LPS in their bloodstream, even if they have leaky gut, because LPS is a very short half-life in the bloodstream. So it doesn't live there very long. So if you fasted for 12, 15 hours, the likelihood that you're going to find any LPS in your bloodstream is very low. But if you do find it, then that means you have a significant amount of LPS that's spilling into your bloodstream on a daily basis. So what he does is he just monitors these students coming into his lab, and he takes the ones that are showing high levels of LPS on the initial testing. And he keeps them, they're his test subjects. And so then what he does, he has them come in fasting, and he feeds them a high-fat, high-caloric meal. In our study, he fed them a McDonald's breakfast. And so what happened after he fed them the McDonald's breakfast was pretty interesting. So when he came in, there was very little measurable LPS in their bloodstream. That's at the 0 hour. At 3 hours, there was a little bit more, but at the 5 hour mark, he saw a 5 to 6x spike in LPS. Okay, so this is the model. So we send the students home for 30 days with nothing but the probiotic, no change in diet, no change in lifestyle, nothing. And these are college students. We know they're not doing anything healthy for their microbiome. Give them a 30-day supply of the probiotic, have them come back on day 31, and have them repeat the test. And we totally blunted the amount of circulating endotoxins in their blood in 30 days. So this was just a pilot, right? We did see some interesting things also, though, in the pilot. We saw a reduction in ghrelin. Now, let me explain this real quick. So the blue area was the placebo group. It was the group that did not have the probiotic. And so what we see is that their ghrelin levels really didn't change over time. But in the postbiotic group, what we see is that there's a reduction in ghrelin. What that means is that the people who didn't take the probiotic, 5 hours after the meal, they're starving. Their body's telling them they need to eat. Now, we understand scientifically why that is. They ate a McDonald's breakfast. They ate something that lacked any nutritional value. So their brain is telling them, you got to eat something. You got to eat something healthy, right? But in the patient who took the probiotic, what's happening is that there's better communication between the gut and the brain. The brain saying, you didn't eat anything. There's no nutritional value in that. And the gut saying, yes, you're right. I know, sorry, but I got 2,000 calories of food down here. Can we wait a little while before we put some more food in here? So good communication between the gut and brain that you lose when there's an excessive amount of LPS in the bloodstream. But when you reduce that, that communication returns. Hugely important for weight loss, hugely important for just health in general. We see improvements in insulin levels. We don't even need to talk about that. We know how important that is. Improvements in glucagon. So the improvements in glucagon and insulin combined show improvements for people with blood sugar handling problems. And we see a number of different inflammatory markers that are improving. So from this pilot, we said, okay, we need to ramp this up. We need to do a full-on study. So we did. And so this study was published in the World Journal of Gastrointestinal Pathophysiology, and it was published as a frontier paper. What a frontier paper means is it means that we've had this problem, we've known about it for a long time, but we've never had anything that can treat it until now. And so what they were talking about is this endotoxemia, this metabolic endotoxemia, what people in functional medicine have been calling leaky gut for the last 30 years. And so it's the spilling of bacteria and bacterial waste products into the basolateral circulation after a meal. So what we did is we studied this and we got the study published. So in the study, the treatment group had a 45% reduction in the amount of LPS in their bloodstream in 30 days. But the placebo group had a 32% increase. So what this shows is that the leaky gut is actually a progressive condition. It builds on itself over time. And if you don't do anything to improve it, it's going to get worse over time. So we're seeing over a 60% difference between these two groups. So I'll go on to tell some of the other things, but because of this, we're actually repeating this study right now. We started it pre-COVID, and as you can imagine, it got blown up because the students couldn't come back into the lab and yada, yada. But so we've restarted it back up about 2 months ago. We're hoping that we'll have the study finished by the end of the year and we can get it published next year. But what we're doing is we've doubled the amount of people in the study and we've tripled the length of time of the study. So we're doing a 90-day study instead of a 30-day study because we believe that we're going to see a much wider discrepancy between the two groups because it looks like this placebo group that the leaky gut is building on itself. So we'll be excited to share that information with you, hopefully by the end of this year. So one of the main things that we saw in the full study was the reduction of inflammatory cytokines. The reduction in these inflammatory cytokines, which were at the root of all those conditions that we spoke about a few slides back. But don't forget, we're in COVID times. And COVID, whether we're talking about the actual infection itself, or whether we're talking about the long haulers who don't have their sense of smell or taste 6 months later, fatigued and still showing symptoms, even though the virus is gone. So if we're talking about those two groups of people, what we're talking about is a cytokine storm. We're talking about the same type of inflammation that's associated with the leaky gut is the same inflammation that causes someone with COVID to go from, feeling a little bit run down, feeling a little sick, to being on a ventilator or dying, right? It's that inflammatory cascade of cytokines that's responsible for that. And we're showing the ability here with probiotics to reduce that. So if you've got people with active infection, or if you've got a long hauler, that's what people like us are going to be seeing in our practices over the coming year and a half. We're going to be seeing these long haulers who aren't getting better, who are looking for anything to help them get better. And so what we're seeing in our offices is that by restoring the balance of their microbiome, by reducing the initial cytokine reactions that are happening as a result of their leaky gut. By reducing that, we allow the rest of the immune system, the rest of the body to recover from the COVID infection. And so just wanted to drop in a little bit of COVID because that's the world we're living in right now. But that was one of the best findings that we saw in the study, was our ability to reduce those cytokines. We also showed the ability to reduce triglycerides, in this case by 30%. But in this case, these weren't patients that had terribly elevated triglycerides. So let's talk about that. Why? Okay, well, what is a triglyceride? Triglycerides are produced by the liver in response to stress. That's what triglycerides are. And so what we're seeing here is that when we reduce the amount of circulating LPS in the bloodstream, when we reduce the inflammation associated with that, the liver relaxes. It dials back the production of triglycerides because it doesn't need to. There's not as much inflammation in the area. So we did this same study. We did it in people with elevated triglycerides. That just published early in 2020. I'm sorry, late in 2020. But that study showed the same thing. We were able to show that same 30% reduction in patients that had elevated triglycerides. So it's a unique thing. Cholesterol is something that we have drugs for, but triglycerides are something that's hard to treat. And it's not something that medicine necessarily has a good treatment for. So by correcting the microbiome, we can reduce the stress on the liver and reduce the amount of triglycerides it's producing and improve cardiovascular function. Pretty cool. So to review, our goal is to create a diverse forest in our microbiome. Now, my feeling is I'm not smart enough to tell the forest who's supposed to be there and who's not. So rather than throwing seeds into the forest of particular species that I think should be there, instead, I'm going to send in the gardener. I'm going to have the gardener go in, pull some weeds, till the soil, create an environment that's conducive for the forest to dictate who's going to be there and who's not. And so that's what spores do. So reseeding, we've shown there's questions of survival, adhesion, diversity, and harmful, and whether it could be harmful to the organism if it truly did do what you're saying it does, right? We can't reseed the microbiome. What we can do is we can use specific lactobacilli, specific bifidobacteria to create a change to a symptom. We can do that, but we can't change the microbiome with those bacteria. Spores are the gardener. They're designed to survive. They're able to eliminate harmful organisms, able to recondition the environment in the favor, to help favor and balance out the good guys. It detoxifies the environment and improves the growth of beneficial flora. So that's the difference, and that's the science of what we're doing with spores, right? And in cases, in many cases where we're looking at a patient, so if we look at a patient where we're talking about someone who comes in, and I had this happen about a month ago, where a woman came in, she had some GERD, she had some digestive related symptoms, but she also was having what I would consider to be chronic urinary tract infections. And so she was having them with regularity. Every menstrual cycle, she was having 1 to 6 days of urinary tract infection that she was having to take antibiotics for. And so what I said to her was, listen, your microbiome needs to change. We need to get your microbiome to shift to a different status. It's unhealthy. The years of antibiotic use have destroyed your microbiome. We've got to restore health to your microbiome. While we're doing that, I need you to go to Whole Foods. I need you to get some Lactobacillus rhamnosus GG. It's very good at helping you monitor the symptoms of the urinary tract infection, so you don't need the antibiotics while we're taking the 3 months to recondition your microbiome and change it, right? So that's how we want you looking at the big picture of the microbiome. We want you to be able to have a tool where you can say to somebody, look, your microbiome's a mess. We need to recondition it. We need to change it. And that's going to make all these other symptoms go away. If you got a patient who comes in with Hashimoto's disease or rheumatoid arthritis, listen, you've got a lifetime of LPS that's spilling into your bloodstream that's created this autoimmune disease. We need to stop that process right now. And so spores are the only probiotics that have shown the ability to stop that process. And so I'll open the floor to questions now. Happy to answer any of your questions and go from there.
Thank you very much, Dr. Bayne. That was very, very precise and exciting way of demonstrating how the spore probiotics might be the future of in the treatment toolbox as we have as practitioners. Actually, there hasn't been any questions coming up, and we've also one came up, but I have put a few down during your talk, and now more are coming in. So a question is, you recommend 2 capsules per day?
Yes.
In one go, or 1 twice daily, or?
No. So, and we found this out trial and error, right? This isn't, if I was talking to you 8 years ago, this would be a completely different conversation probably. But as it is, we've learned clinically that there's a couple things that the spores do, right? We talked about competitive exclusion of infection. We talked about nutrient production. You know, those are all really important aspects. And if you start with a quarter of a capsule, you're going to get those benefits. But the immune modulation benefits that we talked about early in the lecture, where we were getting that stimulation of the Peyer's patches and the gall, that doesn't happen until we get above 3 billion. We don't know why that is, but we know that that's the fact. So until you get to that second capsule, you're not getting the immune benefits. You're getting the competitive exclusion benefits, you're getting the nutrient production benefits, but you're not getting the immune modulating benefits. So what we tell people to do, and this is my experience, I have thousands of patients on the spores, and I consult with doctors around the world on a weekly basis. I've seen it all. So this is my take on things. If you give this product to 100 people, 10 are going to have what we'll call an uncomfortable reaction, 10 to 15. Of those 10 to 15, 8 to 12 of them, the symptoms are going to be mild and something they can work through. A little bit of a loose stool, a little bit of some intestinal cramping, something that they recognize, but not something that they can't fight through. But that tiny, small percentage of people, those are the patients that keep you up at night, right? Those are the patients that show up where they get extreme diarrhea, they get intense intestinal cramping. It never happens on the first dose. If somebody tells you they took half a capsule of Megaspore and they had this huge diarrhea, it's in their head. That's not true. But if someone says, hey, you know, I was taking half a cap for like maybe 4 or 5 days and I started feeling like not really good, that's from the spores. The negative impact doesn't happen on the first dose. It happens after successive dosing, 4 to 8 doses. Then they start to see some cramping, some loose stools, stuff like that. So what we do across the board in order to mitigate the challenges some patients have is we have a titration schedule. I tell everybody to take 1 capsule every other day for a week. Then take 1 capsule a day for a week. And then take 2 capsules. All supplementation is with or after food, because we've been able to show that the spores function better when there's protein and sugar in the environment. So all supplementation is with or after food. Again, that's different from what other probiotics are saying. And when you take the 2 capsules, the 2 capsules have to be taken at the same time in order to get the immune benefits. So long answer to your question, but I think I probably answered a couple questions there.
Super, yeah, you could probably read the questions as well, if you check the chat. So another question is like, what for children? Can children, I mean, we can give them to children?
Yes, the short answer is yes, okay? We can give it to children, we can give it to pregnant women. There are no downsides to it, okay? What I would recommend though is you follow the titration schedule. So I have let me think how old she is. She's got to be 17 months by now. I've been treating her since she was 10 months old. She had full body eczema, right? And so she, at 17 months old, takes full adult dose of the probiotic and the prebiotic daily, okay? I also have a 10-year-old also with eczema, not full body, but a significant patch of eczema on his body. He can't tolerate more than 1 capsule a day, right? To this point, I've only seen him for 3 months. And so there is no one size fits all. And that should be in everything you do, but it's definitely in this, okay? You should titrate everybody up. If they have an intense reaction, if they're taking 1 capsule every other day, and by the 3rd day, they're complaining of intestinal cramping or diarrhea, scale it back. I have patients who come in who are that kind of canary in the coal mine kind of patient. They go to every doctor in town. They're sensitive to everything. I start them with a quarter or an eighth of a capsule every other day. Everybody has a starting point. You can find where their starting point is and slowly build them up. People that are very sensitive, they appreciate your recognition of them being sensitive and starting them with a lower dose and going slowly. But that's how you want to monitor people. So the short answer is I have children on the full adult dose and I have children on half a capsule every other day. They're getting the benefits. They're getting some benefits when they're taking it. And so you just have to work through them. If somebody can't tolerate more than half a capsule or a capsule a day, then the workaround is to start using the prebiotics and the mucosal repair products to get their microbiome more primed to be able to tolerate more of the spores. So follow the titration for everyone, but everyone can take it. There's no condition that is off limits with spores.
So there's the answer to the pregnant woman.
Yes. And so just open the capsule up. It has no taste. So I have moms with C-section babies. I have them open the capsule up, put their finger on the powder, put the finger in baby's mouth, put the powder on it, and then put them on the boob and have them breastfeed that way. And so that each time they're breastfeeding, they're getting, maybe an eighth of a capsule into their digestive tract. And I'll have them scale it up slowly, but that's how I have them do. That's how I have C-section babies. That's how I have mom supplement with them. And then I come in and I say to mom, well, how much do you think you're giving a day? They're like, well, I don't know. I do that 4 or 5 times a day and I just give it to them. And that I don't know exactly how much it is, right? That's okay, right? You're supplementing the spores and they're seeing improvements with the baby. So you can just open the capsule. It doesn't have any taste or smell. It's very easy. You can pour it on food. You can pour it in a drink for a kid and have them drink the drink to get it down. So very easy to supplement.
So there is this discussion around treating SIBO, whether you should treat with only antimicrobials and not treat with probiotics at the same time. What's your point on that?
Well, if the question is, if you're asking the question of should I treat it with lactobacillus and bifidobacteria probiotics, I would say absolutely not.
With your product.
Yeah, but spores are okay, and here's why. So lactobacilli and bifidobacteria in the SIBO patient, because that bacteria is overgrowing in the small intestine, there's a histamine sensitivity. There's an increase in histamine production, and there's an increase in the sensitivity to histamine, right? And so lactobacilli and bifidobacteria can be histamine inducing. And so taking a lactobacillus or bifidobacteria-based probiotic can definitely irritate a small intestine bacterial overgrowth. But now let's talk about the spores. So if you remember, I told you, and I did it on purpose because I knew you guys were going to ask me a question about small intestine bacterial overgrowth, but when I explained it, the spores resporulate in the lower part of the small intestine, right? So there's 2 types of small intestine bacterial overgrowth. One is I ate a bacteria and it's now living in my small intestine. If you've got that kind of small intestine bacterial overgrowth, the spores will kill it 100% of the time, every time, okay? The only problem is that's a very rare type of small intestine bacterial overgrowth. You almost never see that type of SIBO. The type of SIBO that we see day in, day out in our practice is when beneficial bacteria creep into the lower part of the small intestine. Could be because of problems with the ileocecal valve, could be for a number of other different reasons. But the small intestinal bacterial overgrowth is predominantly living in the lower part of the small intestine. As I told you, the spores resporulate in the lower part of the small intestine. So there's no competitive exclusion of those bacteria. There is the immune modulating benefit, and that's ultimately what's going to correct long-term the small intestine overgrowth, small intestine bacterial overgrowth, is that immune modulation, that improvement to the immune system is what you need long-term, but you're not getting any short-term competition with that bacteria to get it out of the small intestine. So you still need antimicrobials when you use spores, but spores are by far the best probiotics that you can use. One, because they don't make matters worse. Two, because they're improving the immune function, which is ultimately at the root of why you have that SIBO to begin with. So short-term, you need the antimicrobials, but long-term, you need that immune modulating benefits that only the spores will give you.
So your protocol, and now it's because I know there's published research on that was on rifaximin, which when we would perhaps, a group of us would perhaps prioritize using herbal antimicrobials or more natural remedies. So would you use a period where you only use the antimicrobials and then you add the microbiome products after that, or just start with both? Of course, titrating up.
We have research, and it was a different study, but we did a study on hepatic encephalopathy. 100% of the patients in the study were on rifaximin. And we showed the ability to function, that spores were functioning in the rifaximin environment. Now, I don't believe that rifaximin should be the first choice of treatment for small intestine bacterial overgrowth. I'm not saying that. But my point is that the spores function in an antibiotic-rich environment. They make antibiotics. So if they were killed by antibiotics, then it doesn't make sense, right? They're producing antibiotics. They can produce antibiotics. So what we're saying is that the spores are your best opportunity to improve your immune function. And they could care less whether you're taking an antibiotic or whether you're taking a natural antimicrobial. It doesn't impact them. So the only thing that we've actually shown a natural ingredient that is bad for the spores is essential oils of lemon and wormwood. Those are the only natural products that we've been able to show that disrupt the function of the spores. So beyond that, everything else doesn't really impact them in any way. So if you're using oregano oil to get rid of the small intestinal bacterial growth, there's no problem taking the garlic, no problems at all. Nothing besides essential oil, lemon, and wormwood. We've tested everything.
Okay, super. That's good to know.
Those two.
All right, I think that was the last saying for today's event. Thank you very much for your time. Pleasure. And
listen, I'd like to add one last comment, right? So this is a paradigm shift in thinking. And so people respond differently to a paradigm shift. Some people crawl into the corner, get into the fetal position, and question everything they've ever done. Please don't do that, right? What we're trying to do is we're trying to use science to understand how to improve the things we do, right? And it's called practice for a reason. We're learning as we go, right? I've used all types of products. I don't have anything bad to say about any one product, but what I want you to understand is that really looking at the science of the microbiome, really understanding how these bacteria function together and how to improve the microbiome long-term, it's very important to have spores as part of that reconditioning process. That's all I'm saying. I'm not a know-it-all. I'm not saying that this is the only way to do things. I'm just providing this information, and I hope that it brings questions. I hope it stimulates a conversation. I hope it brings things where you'll try new things and use a critical eye to test them and to see if they're if they hold up to what I've told you. And so that's all I can really ask. And I think that once you get using the products, I think you'll find that that the products work very well, and that you'll be happy with your clinical outcomes.
Super. Thank you very much.
And thank you for your time. I appreciate it.
Thank you, everyone. Remember, the recording will be shared with you in Monday or something like that, as soon as possible. So, and the slides and so on will be shared with you as well. So I hope to see you next time with Graham. So until then, take care. And if I don't see you, then take care anyway. Bye.